So, and thank you for the kind invitation. So I'm very happy to give this lecture, and both to give the lecture, but also to express my support for all the Ukrainian people in this very difficult situation. And while I think many of you may have more important things to think about these days, I still hope that it can give you some inspiration in between, and that by recording the lecture, you can pick it up later. And I also hope that this can contribute to maintaining good science and developing science in Ukraine. So what I will do has some overlap with what you heard from Edvard Moser about 2 weeks ago, but it also has a somewhat different perspective. I will provide you with some history of brain science, and particularly science of location, how we find our way and how we know where we are, and how this relates to our emerging understanding of computation in the brain, how neurons in the brain compute. So let me start first with a question. So the first slide here, this is probably a picture that you also saw from May-Britt, but it illustrates the goal of our common lab, which is to understand how the brain knows where we are. Where are we in space? Why am I? How can I know that I'm here and not there? So this is a psychological function that is absolutely critical for animals and for humans to survive. We really need to know where we are if we want to find food, to find a mate, and to survive in all kinds of ways. The sense of location is absolutely central to the brain, and for that reason is developed in all animals. But it has some advantages because it is expressed in all animals, and also very much apparently in the same way in all mammals. We can study it in simpler animals, laboratory animals, and thereby still understand quite a lot about what's going on in the human brain. And what our hope is that by understanding the sense of location, we'll actually understand much of computation in the brain in a more general sense, because often the principles that the brain works after are much the same, quite often the same in different brain areas for different purposes. So let's go back in history, maybe about 100 years. So during the first half of the previous century, there were several psychologists, and particularly a branch that developed into what was called behaviorism, mostly in the United States, because this was after the Second World War, and the United States was the place where most of the development happened around and after the Second World War. So what happened in the development of psychology there was experimental psychology, was an attempt to understand or to identify the real elements, the smallest pieces of behavior. So these psychologists, and 3 of them are mentioned here, Watson, Hull, and Skinner, tried to break down behavior in what they called stimuli and responses. Stimuli elicited a behavior, was something in the environment that caused the behavior to happen, and responses were the animals or the humans' response to the stimulus. And then they broke that down and described behaviors as chains of stimuli and responses that happen in a sequence. So this was, of course, extremely simplified, but it still had some success. And because if you describe behavior as stimuli and responses, you could actually, and then I should mention there's also a third factor. reinforcements or rewards will happen after the response. So if an animal or human does a response or a behavior, and this is followed by some sort of reward, then that response becomes more likely. So for example, this is still used very actively in dog training. So if a dog, for example, raises its paw and then immediately gets a dog treat, then the dog will continue to do this over and over again. And by using these principles, the psychologists then developed behavioral science where they could actually train animals to do the weirdest things. And they just show you one thing here then.
Two pigeons are at either end of a small ping pong table. One pigeon pecks the ball as it comes toward him and knocks it toward the other pigeon. Other pigeon pecks the ball
so
back
these
across
are
the table.
pigeons playing
if it
ping
goes
pong.
past one pigeon, the other pigeon can
And this
eat,
is maintained
and if it goes the other
by
way, the other pigeon eats. So
pigeon getting a reward
real game.
each time it manages to hit
pigeon,
the ball out on the other
is
one's side.
reinforced for a
Now this one
cross-court
gets a
shot
reward.
if that is what gets the ball past his opponent.
So I'll stop there, but I mean, by giving rewards at the right times, you can make animals do anything. And these psychologists then thought that this was the basis of behavior in all kinds of ways, but still they had to describe behavior as chains of stimuli and responses and rewards. And of course, this was far too simple. And one of the people who responded or reacted to this simplification was a psychologist called Edvard Moser. So he had a more global view of behavior. So he suggested that knowledge is based on more on maps in the brain. And he suggested that when animals find their way around in a maze or anywhere in a house or wherever they run in the forest, they create a map of the environment, and then they store information about locations and about their experiences within this map. And he then made different mazes. Rats run in the mazes, started one place, ran to a goal, and he showed that if he changed the maze that the rat had been running many times, and suddenly made different arms, it will always choose the arm that goes most straight to the goal place. So in that case, it's the location shown as P here. So even if the rat had never run this path here, it would choose it because it was the quickest route to the goal. And that suggested to Tolman that they must have maps that help them find their way. So, but Tolman didn't know anything about the brain because there was very little known about the brain at this time. This was in the mid-1950s. And today we know a lot more about the brain, and we know that these maps probably include parts of the brain that are called the hippocampus and the entorhinal cortex. So you see the hippocampus in red here and the entorhinal cortex in blue. So both of these are in the medial temporal lobe, that's on the side of the brain, somewhat behind your ears and somewhat buried under the neocortex. So what do we know about these brain areas? Well, I'll take you into that, but let us first explain why most of the work in this field is done not in human brains, but in animals. So we have the human brain here, which has approximately 86 billion neurons. So it is just a lot and very difficult to understand, and of course also very inaccessible. But then as we go down in evolution, passing monkeys, getting to rats and to mice, the number of neurons is getting more manageable. So in the rat, only 200 million, and in the mouse, even just 71 million. So, and you can continue even further to insects or, and the smallest organism studied for brains is the nematode C. elegans, which only has 302 brain cells or neurons. So one can understand quite a lot about how brains work with working simpler brains. And the reason is, of course, evolution, because every brain is thought to evolve out of a simpler brain of some common ancestor. So much is being shared between rodents, which we study most of the time, rats and mice, and for example, humans. So, and that applies also to the organization of the brain. So this shows a cross-section through the cortex, the neocortex, which covers most of the brain's surface, and which is what we think of as the site of much of the intellectual activity of the brain. So in humans and in other mammals, it is 6 layers in most brain areas of the cortex, and you call them 1, 2, 3, 4, 5, 6, and then there are sublayers in many species. So this just shows that the organization, the way the brain is built up, is very much the same in different species. And the same comes to the way that the brains actually work, because brain cells or neurons work by emitting electrical signals. So these electrical signals, they run through the neuron from one end to the other, and then out the neurons have branches, and these branches then carry these electrical signals until the signal comes to an end of a branch, and then it's passed over in the form of chemicals to the next neuron, which again transmits an electrical signal. This was discovered about 70 years ago in squids. So this is because, and the reason is that at that time they were much easier to study because they have huge nervous systems and very large cells, so they could be discovered. It was shown that the electrical signaling mechanisms is electrical and in the form of what we call an action potential. This is the unit of electrical transmission, a certain voltage that is carried through the cells in order to communicate signals. So how can we record the activity from these signals or from these neurons? Well, this is by electrodes. So these are very, very small electrodes, microelectrodes, which are thinner than a hair, which can be put in between neurons in such a way that they don't damage the neurons, but can capture or pick up the electrical activity from the voltage changes from the neurons that are very, very close to the electrode. And these were developed in the 1950s. And at that time, it was often in the form of a tungsten wire, a wire that was very sharp at the end, and then could pick up the electrical activity at the end, which was just a few micrometers wide. So small enough to pick up signals only from a single cell or possibly a single cell and its very closest neighbors. So that brings me then to the early 1970s. So this was the year when John O'Keefe, with whom we shared the Nobel Prize in 2014, this was the year when John started recording and from a brain area, which I showed you, the red area here, which is called the hippocampus. So he recorded not in the human brain, but in a rat brain, and had electrodes that were squeezed in between the neurons and could pick up electrical signals from this brain area. And then through a very thin wire, this was carried to a computer and an oscilloscope, and it was possible to see when an individual cell was active and showed these electrical action potentials. What he found is what I'll show in the next slide, which you probably also saw in a slide from Edvard Moser's lecture 2 weeks ago, but I'll show it again. So what you see here is from above is a rat. The rat is running around in a box. So this box is 1 meter by 1 meter. And the rat is running around because there are small chocolate crumbles around in the box. And the rat picks them up. And this is just a trick we use to have the rat visit every possible place in this box. And then at the same time, we are recording the activity from one single cell using these very, very small electrodes that are inserted into the hippocampus. So let me then start the movie. We will listen and see the the activity of one single cell or neuron. And each time the cell is active, you'll hear a sound, and you will also see a red dot appearing on the display. So these are the sounds from a single cell. Each time it's a bop, then it means that there is an action potential in the cell. You can see that this cell is only active when the rat is in the upper left part of this box. Otherwise, the cell is silent. And because this cell was only active in one place in this environment, in this box, and John O'Keefe called these cells place cells. They're active only in certain places. Can also be shown in a color version here. So hot colors, red, means that there is high activity, and blue means that there is low activity. And different cells had different place fields. So one cell might be active here in the upper left part. Other cells might be active in the bottom right, or in the middle, or in the left bottom part, and so on. And imagine then that there are thousands of cells of this type in the hippocampus. So that led then John O'Keefe and Lynn Nadell to suggest at the end of the 1970s that these place cells in the hippocampus are actually a part of a map, a spatial map in the brain. And in that sense, they provided some brain mechanism for the map that Tolman had proposed about 3 decades earlier, when he suggested from his maze experiments that rats and humans actually must have internal maps. He just couldn't say where and how, but these place cells suggested that there are cells that, by being active in different places of an environment, together can tell the rest of the brain where the animal is at any given time, because the combination of active cells is different at every possible place in the environment, and then it's possible to read out from that activity where the rat is at every given second of the experience. So then this turned out not only to be the only type of spatial cell in the brain, because some years, actually 10 years later, or even 20 years later, it was discovered that there are also some other type of cell, which are directional cells, cells that are active only when the rat was pointing its head in a certain direction. So it could be forward, could be to the side, or to the other side, and so on. And what you see here, so these cells were discovered by Jim Rank and Jeff Taube, who were working in a lab outside New York at that time. And here shows an example of that, such a cell. This is one single cell. On the x-axis, you have the direction of the animal. So this is just relative to the room. And then the activity of the cell is on the y-axis. And you can see that this cell is not active in one given place, but is active in a certain direction. And that direction for this example is around 300, 250 degrees. And these are just in this area of the brain, which is very close to, but different from the hippocampus where you had the place cells. And then another 10 years passed, and this is where we came into the picture. So we recorded again from a third brain area, which you see up here. So this is a rat brain seen from behind. And the colored area here is what we call the entorhinal cortex, is what you saw as a blue area in the human brain that I showed you previously. So in this brain area, which had not been investigated much at all when we started with this in around 2005, then if you start the recording activity there, this is what you get. So rat is still running around in the box. And each time the cell is active, you get the white dot in this case. And you can see that this particular cell, is just one cell, is active also at certain places. But those places aren't only, they are not only one place. There are many places, and they form clusters. and those clusters seem to form a very regular pattern. So this pattern can maybe be seen even more clearly in the picture to the bottom right here, where in gray, this is again a box, but now it's a bigger box. It's more than 2 meters each direction. And in gray, you see the trace of the rat as it was running around in the box. And in black, you see the activity of that particular cell. And you can then see that it's different from the place cells in that this cell is active not only in one place, but in many places, but also that those places form a pattern. And you can see the pattern. If I now put these triangles on top here, you can see that it's a triangular pattern that repeats itself all over the space. So actually it is hexagonal. So the repeating unit here can be considered as a hexagon that then repeats itself. So in many ways, this can be seen as a grid that covers all over the space. And for that reason, we then call these cells grid cells. All right, so let me then just say that these exist at different sizes, beginning at the top of the brain here with the small grids that are little distance, and then you get bigger and bigger grids as you go deeper into the brain. And these grids form different clusters that then have quite overlapping, so they're distinct different maps. There are small maps, and there are medium maps, and there are big maps, just like you have maps for different sizes of the external world as well. So let me skip that one. Just then say that these different types of cells, such as place cells and the directional cells and the head directions, the head direction cells and the grid cells, and also other types of cells that May-Britt mentioned, namely border cells and so on. All of these have been found in several species, so it's nothing special for rats or mice, really. They are also present in bats, for example, flying bats. And these flying bats, they are on a completely different branch of the phylogenetic tree compared to rodents. Although they may look similar, they are not really. And they have also been found in humans. So in humans, you can discover these cells because occasionally patients with severe epilepsy need to have electrodes implanted in the brain in order to find where the epileptic seizures start. And then you get for free the activity from the human brain when a person moves around. So in most of these cases, the person doesn't really move, but sits in a bed and then looks at the computer and they use virtual reality to simulate movement in space. But you do then find cells that have these patterns as well. So let me then move to today's work. So what I showed you now is the work that we and others did during past decades and with the key discoveries happening around the year 2005. But today, neuroscience or brain science has developed further. And what we and others now are working on is to try to understand how many cells work together, not seeing only what are the properties of single cells or individual cells, but actually seeing how thousands of neurons work together to create a sense of where you are. And this was not possible when we started out in the field, but very recently, new techniques have been developed that make it possible to record activity in mice or rats when they run around from thousands of cells at the same time. I show you one here. These are the so-called neuropixels probes, which essentially is a computer chip. And the chip is the very, very thin wire that you see here. It actually is a computer chip like you see to the right here with thousands of dots spaced across this probe. The probe goes down into the brain with the bottom here. And then it is possible, because this probe is so thin, it's just 20 micrometers wide. it is possible to use every single dot here to pick up electrical signals that are from around. And this is very different from the old way it was done, because then only the tip could be used to actually pick up those signals. So suddenly you go from picking up electrical activity from maybe 10 cells at best at one time to many thousands. And with these probes, you can then get not only one picture of how a cell was active, but this just shows 149 grid cells. So each square here shows the activity of one grid cell in a box. And you can see the grid pattern for the different cells. And this is just 149 out of a total of 2,460 that were recorded. And you can also display them in a different way. So this shows actually 149 lines. So each line is one cell. And whenever there's black, the cell is active. Whenever it's white, the cell is silent. And time is then on the x-axis. And then you can make slices, like the red column here, through bits of time, which, for example, could be 100 milliseconds long. And you can analyze the combined activity of these cells. So this gets very difficult and then needs complicated mathematics to understand how the combination of activity evolves over time. Because if you have, let's say, only 149 cells and they vary independently, you can plot the activity, the combined activity, in a 149-dimensional coordinate system. But of course, we humans can't really, we're not made for watching activity in a 149-dimensional coordinate system. It's just too complicated. But we have mathematical techniques to break down the activity into the three most important axes into a three-dimensional coordinate system. And then we can, which then still accounts for as much of the variation as possible. And then we can see how the activity moves through this three-dimensional coordinate system instead of the 149-dimensional one. And the funny thing is that when we do that for the grid cells, we actually see that the activity, which you can see in this trace here, moves around in a cloud that has the shape of a donut, or as we call it, a torus. So you see the hole in the middle here, and like donuts, just like you can eat, right, a donut. And each dot here is one single point of time, and you can see that the activity then moves around on this donut all the time. So on grid cells, this happens regardless of whether the animal is walking around in the box, whether it is sleeping, or doing anything else. And you may wonder, why should the activity move around on a donut or a torus? Well, this is, of course, too much to compress into a lecture, but just want to say that there are theories that are studied in physics. They are called attractor theories or attractor network theories, which is about how cells are wired together. And if cells are wired together in a certain way in the brain, you can actually, by having similar cells connect to similar cells, you can actually get this type of pattern, and you would even expect it. So when we then saw these donuts, it was actually a confirmation, a major confirmation of a theory for how cells collaborate in big networks in the brain. So then let me move to a slightly different question. So now I showed you some of the electrical activity and how we can study it with these very, very modern probes that can pick up activity from many cells at the same time. But we also want to see these cells, right? So for that, and this is was also briefly mentioned, I think, in Edvard Moser's lecture, but I will repeat it here in a slightly different way. So this is about microscopes. So there is a certain type of microscope that allows you to see the activity of cells at the time scale of the cell's activity. And ideally, you want to do this while a mouse or a rat is walking around or doing whatever it likes to do. So what you see here to the right is a picture of a certain area of brain. So this is about 400 by 400 micrometers. And every dot here that you can see is one particular cell. And you can then, usually this was studied by, let me go to the next slide, in one or two ways. So either you had mice running on a ball in a fixed position, so that it's like being in an MR scanner, but an MR scanner type of equipment for mice. So they were fixed with a head so that they couldn't run. And then because the head could not move, they could use a microscope to look down onto its brain, a certain small area of the brain. And then they compensated for the fact that the mice could not move by having them run in a virtual environment. So it's like a cinema where they play the movement in space. But the disadvantage of this is, of course, that it's not quite a real movement. It is, it's kind of fake, and quite often the animals detect that. So a different way of doing it is what has been called one-photon microscope, which were so small and are so small that they can actually be carried on the head of a mouse. But the solution that has existed until now is based on one photon solution, where you use, in this case, just actually use a kind of camera to take a picture of the activity of the cells. And you can do that because the cells express a fluorescent molecule that is activated each time the cell is active. So you can get a fluorescent signal, and then you can see the cells being active. But the problem with that type of microscope is that it has very low resolution and picks up a lot of background noise from other cells. So for that reason, we then tried, or people have tried over 20 years to develop a so-called two-photon microscope, where you actually use a laser to activate a point that is reached only by those two lasers. Then you get, with the laser light, you activate the cells, and you can actually monitor the light that emits at a much, much higher resolution. But making this so small that it can be carried on the mouse head has been very difficult. And 20 years have passed, and it's either been too heavy or too slow, or cables were not stiff enough, and all these things, so that it's only now that we have a microscope, which is Wei-Jan Song's development. He is a postdoc in our lab. This is just 2.4 grams and can be carried on the mouse head, and it has all the equipment that is needed. Laser light comes in at very high precision, and the light fluorescence is picked up, and there's a scanner that goes all over every possible part of the picture at a very, very high speed, so that you can actually get a movie of the activity. And this can be done then while the mice run around in the environment. And what you see here is 3 different mice. To the left column, you see a normal mouse with nothing on the head. And this, in the middle, you see a mouse with the new microscope on the head, and you shouldn't see much of a difference. And to the right, you see the old versions where the mouse very soon gets tired and just sits in a corner and doesn't really want to run. So this is a big improvement. And I'll just show you then some movement here, where you see the activity of a window of 400 by 400 micrometers. And each blinking place here is a cell. And this is recorded at the same time as a mouse is running around in the box. And this camera is at the bottom so that it can pick up the electrical activity of the mouse when it's running around in the box. And at the same time, you see the activity of the cells. And this can be done at 4 different planes at the same time, at 4 different depths, because the scanner is so fast that you can do this actually at 40 hertz, 40 times per second. So that means 10 seconds per each of these images. And you can actually reconstruct the activity of the cells and do this for more than 1,000 of cells in this volume of brain tissue at the same time. All right, and finally, just want to show, which is probably also what you saw in Maibritt's lecture. This can be done in whatever behaviors. So this is a mouse that is running just vertically on a 20-centimeter high tower, climbing up, getting a biscuit, which it likes very much. And then at the same time, you have the activity in the brain. Here you see the cells blinking. And now you'll see the mouse jumping down and then doing it again and going up and getting the biscuit and then doing this over and over again. And the reason why we use this task is that when the rat is jumping, particularly, it stretches the cable. And this is when you would expect to get movement artifacts in the recordings. But you can see that the activity is really the same. It's not much. And even if you do, there are computer algorithms that can make you correct the motion artifacts. It is still very little. So you can get very accurate recordings of the activity of the cells with this new microscope that was just published actually a few weeks ago. All right, and this just shows you how you can record many grid cells at the same time. And you can then actually map the activity of cells. So this shows for entorhinal cortex. Grid cells are the cells that are shown in red, and other types of cells are shown in blue here. And with this microscope, you can then describe how different cells in a brain area work together. So with that, then I want to now at the end just say a few words about the uses of this, because what I've told you so far is basic neuroscience. It's basic science where we try to find out how the brain works. But nonetheless, it is also of use for different purposes. And I will mention two of the uses. One is for health reasons, and the other is for computational reasons. So let's begin with health. So we believe that by understanding the cortex, the cover of the brain, which is responsible for our intellectual activity, we will eventually understand what's going on sometimes, what's going wrong sometimes in this part of the brain, which give rise often to neurological and psychiatric diseases. And such diseases of different kinds, they could be anything from addiction to dementia, to epilepsy, to mood disorders, depressions, and so on, schizophrenia. All of this actually affects up to 1/3 of the population. And quite often it is related to activity or abnormal activity in certain brain areas. And by understanding how the brain works and how it computes, one can actually, hopefully, eventually understand how these diseases emerge. And this particularly involves, in our case, the development of Alzheimer's disease or dementia. This is a major type of dementia. And the reason is that this brain disease, which affects so many people and currently more than 50 million people in the world, and is expected to increase to more than 150 in 2-3 decades because the population is getting older and older. So so this just shows the development with age. So particularly above the age of 80 to 85, as we all know. But what's particular about this disease is that very often it starts with cell death in the entorhinal cortex, which I told you about, which contains the grid cells, and also in the hippocampus. And the result is that the persons who are affected by this can't find their way, and they get problems with memory, which are two brain functions that depend on those areas. And very often, this disease starts a long time before symptoms of Alzheimer's disease emerge. Quite often, even a decade or two, you can see that this brain area starts shrinking. All right, and the second point I wanted to make is that there are reasons for understanding brain computation that are not only related to health, and that is to understand or to develop better computers. And while computers have been very good at finding information very fast and doing calculations and doing serial operations. What computers traditionally have not been so good at is parallel, doing things in parallel, and often in noisy information. That is when the brain is much better. And until just 10, 20 years ago, computers even struggled just to identify an object. For example, if you showed a picture of a chair, then the computer might not actually understand it was a chair because it's a different angle, different light conditions, and so on. Now with AI, this is getting much better, but nonetheless, this is what the brain is good at, namely doing many things in parallel and often with very limited information. So there are many differences between brains and computers, and I can't mention them all, of course, but I want to emphasize one, namely the energy consumption of the brain, which is only often said to be only 20 watt, which is very, very much less than the supercomputers that have been developed for doing advanced computations, which often use many megawatts. And I think that raises an issue about the energy use of computers, which is enormous. And if we were to find out how the brain can do similar computations, or perhaps even more advanced computations with so little energy, then there might be something to learn for the development of future computers. So with that, I will just then start to draw conclusions here. So what is common between modern computers and the brain is the use of networks with different nodes. So by understanding how brain networks work and how they compute, one can actually also understand a material that could be useful for developing better computers. And particularly important here is what is the brain's ability to learn. So this shows two connections between two cells, the so-called synapse, and which are strengthened and weakened, and which we know a lot about the the molecular mechanisms that underlie this. So this is thought to underlie learning. But now there is the developing field of machine learning, which also helps computers to learn from their own experience. And that is already making computers so much more efficient. So I think that is also part of what the brain is good at. It's not just a machine, but it's actually a machine that learns from itself all the time. So then let me just conclude this lecture. But one thing I wanted to say is that the work I presented is, of course, the work of many, many people. So I don't have time to mention individuals here. But now in the end, let me just say that you have all the sympathy from me and my colleagues here, and we want to do anything we can to keep your hopes up and know that that in the end justice will win. We support you, everyone in Ukraine, and I had never imagined that so terrible things could happen now in modern days, but it does happen, and we hope you stay strong, and we support you, and we also hope that it is possible to maintain good science in Ukraine, and that this lecture series will contribute to that, to keep science going and to recover as this war is over one day, hopefully very soon. So with that, I want to thank you for your attention. And I think then I go out of the lecture, and I would be happy to take any questions that you might have. Thank you very much.
Thank you very much, Professor. We have a lot of questions, but I will read out the most interesting one. So, do you expect a big breakthrough in neuroscience in the nearest future, and what will it be?
Yes, I do think there is actually an ongoing breakthrough, but neuroscience has developed enormously during the past years and decades. And I think much of the reason is that it is now possible to record and monitor the activity of many, many cells at the same time. And we know that the most complex brain functions, such as thinking and planning, memory, and so on, they all depend on the collaboration between very, very many cells at the same time. And we didn't have the tools to study this, but now we have them. It is not only about techniques, like I mentioned, microscopes and probes, but it's also about how to analyze them. And for that, you need very, very strong computers because it's a lot of data and you need quite sophisticated analysis. But all of this is now there. So I do think that neuroscience is on the path to understanding the basic mechanisms of cognitive functions. That means, as I said, memory and planning and just having an idea, thinking, and so on, which were quite unimaginable some years ago. And that is perhaps the breakthrough I would think will happen soon.
Okay, thank you. The next question is from Sophia. Many people say that the human brain works only 10%. How can you comment on this statement?
Yes, that is a very common statement, and I would say it is simply wrong. That is, and but I don't know where that idea has come from. It has been alive for many decades, but how would I say that it is wrong? I think that all parts of the brain are active in some way or other all the time. So there's never any part of the brain that we are not using. But that said, many neurons are quite silent much of the time. And I can take as an example the place cells, because if you're not in a particular place, then a place cell is not active. But it is still active when you get there. So I think every part of the brain and every neuron in the brain is involved in some important activity or not. And you can develop this, but because you can train your brain, and it's been shown in animals, in rats and mice, that if you give them good conditions, they learn a lot, they can wander around in big cages and so on, and have a lot of toys, then the number of neurons increases, but it's particularly what increases is the connections between the cells. It gets much richer, many more connections. So I think you have the neurons you use them all the time, but they can become even, the connections and the richness of the network can become even bigger. But this statement about 10% is one of the myths that exist in neuroscience, and it's just not true.
Okay, and the last question is about perspective and future. Do you expect any threats from artificial intelligence?
Yes, so I think that artificial intelligence is, as you know, it has been on under development for many years and has really had a breakthrough. So you can now use artificial intelligence for many things. But I think it will become important also for science. You can use artificial intelligence to find patterns and see patterns that you can't really see with the human eye. And examples are, there's a lot of now talk about using artificial intelligence for understanding protein folding. So this is how proteins fold in space. And there are some ways that are possible, some ways that are not possible, and some are good and some are not bad for making cells do what they are supposed to do. And this is just one example where breakthroughs have now been shown recently, where you actually, it takes a very, very long time to understand how a brain or a protein could fold in a certain way and create some function, whereas artificial intelligence can do it much better. And I think the same comes to the brain. So when you have these complicated patterns of activity among thousands of cells, we often don't see it because it's just so much data, and artificial intelligence might identify patterns. But still, in the end, I do think we'll still for a long, long time need human brains, because in the end, artificial intelligence still just essentially does what it is told to. And you need the creativity of human brains to put it all together. But I think as a tool, it will certainly be very valuable in the future.
Okay, thank you very much for your presentation and for your answers. Unfortunately, we have a very limited time, but thank you very much for joining and for supporting Ukraine and Ukrainian science.
Okay, I appreciate very much, and then fingers crossed, and I hope the best for Ukraine and for all of you.
Thank you very much. Шановні учасники, дякую всім за запитання та доєднання до нашої лекції. На жаль, сьогодні ми дуже обмежені в часі. Дякую всім і чекаємо вас наступної лекції. Thank you very much. Goodbye. Have a nice day.
Okay, goodbye.